A similar understanding of chemical bonds is required in the unit number 1 of CSIR NET syllabus. In point number C, van der Waal, electrostatic, hydrogen bonding, hydrophobic interaction, these are all examples of different kind of chemical interactions or chemical bonds. Although the unit number 1 contains 10 different topics, of which only one topic defines the stabilizing interaction. But if you look closely on the other topics, for example, structure of atoms, molecules and chemical bonds, composition, structure and function of biomolecules, can we study all these things without the understanding of chemical bonds? So chemical bonds is the foundation to understand all kind of relations between the atoms, between the molecules and in the life processes. We cannot understand the folding of proteins or conformation of nucleic acid without the understanding of covalent and non-covalent interactions or bonds. So more or less, unit number 1 is based on the understanding of various kind of molecular interaction. Some bonds involve sharing of electrons, while the others do not. Some bonds are strong, but some bonds are weak. To get a more detailed idea about the bonds, first of all, you have to understand it is not the energy which is the most important part, but the number of interactions involved in a particular type of bonds. We can classify all kind of bonds into subcategories. For example, bonds can be covalent, and covalent bond can be polar or non-polar. Or bonds can be non-covalent. While covalent bond involve sharing of electrons, non-covalent bond do not require sharing of electrons. These can be ionic, coordinate, hydrogen bonds, van der Waal interactions, electrostatic interactions, hydrophobicity and other interactions. Generally, non-covalent bonds are weaker bonds and the covalent bonds are the stronger one. And covalent bonds can be further defined as polar and non-polar covalent bonds. And on the basis of number of electron pairs shared, they can be single, double or triple bond. To understand the importance of all type of chemical interaction, we should be very clear about all these classifications. When we study about protein synthesis during translation, we all know that there are various kind of bonds at different level of protein folding. In primary structure, amino acid joined together by a covalent bond known as peptide bond, forming a primary structure of protein, which is not properly folded. Now comes the secondary structure. In secondary structure, non-covalent interactions like hydrogen bond enforce polypeptide to take very precise and defined physical conformation like alpha helix, beta pleated sheets, turns, loops. These all structures are known as secondary structure, and you have to always remember that secondary structures are dependent on the hydrogen bond between the oxygen of carbonyl carbon and hydrogen of amide nitrogen. Hydrogen bond is a type of non-covalent interaction, a force responsible for the secondary structure of proteins. At the tertiary level, polypeptide folds more. Now comes the interaction between R groups of amino acid within the polypeptide chain. This interaction can be ionic interaction or hydrophobic interaction, hydrogen bonds, or a single type of covalent bond which is also known as disulfide bond. In tertiary structure, most of the forces responsible for protein folding are non-covalent. Only one particular type of covalent bond is present which is known as disulfide bond, and it is not necessary that it should be present in all type of protein folding. Polypeptides, which has sequences that can form disulfide bond, can show this disulfide bond formation, and it is very crucial in protein folding. All of these interactions are intramolecular interaction. If protein folding goes for the next level, that is quaternary level, polypeptides which are locally folded can interact with each other, and intermolecular bonding or interaction can occur. This interaction can be again non-covalent like hydrogen bonding, hydrophobic interaction, ionic interaction, or a single type of covalent interaction which is known as disulfide bond. It is not only proteins which show the importance of chemical interactions, but all the other biomolecules are also dependent for their structure and function on various type of covalent and non-covalent interaction. For an example, double-stranded DNA molecule. In DNA molecule, backbone contains a covalent bond, phosphodiester bond, which is responsible for the polymerization of nucleic acids. Bases interact with each other. For example, adenine pairs with thymine or guanine pair with cytosine by the help of non-covalent interaction, that is hydrogen bonding and hydrophobicity. On the one side, the disulfide bond, a type of covalent bond, is a crucial player for the polymerization or synthesis of DNA and RNA. At the same time, non-covalent bond helps to accomplish appropriate correct base pairing between A and T, G and C. Similarly, in lipid bilayers, two types of interactions are very crucial. One is hydrophobic interaction between the tails of the phospholipid molecules and a small electrostatic or ionic interaction between the head groups. The proteins which are present in the lipid bilayer and are responsible for various roles also interact with the lipid bilayer by the help of hydrophobic interactions or other similar kind of forces. Some special molecules like hemoglobin, 4 peptides are joined together by the help of non-covalent interaction. They also have non-protein part which is known as heme. Heme is attached to protein by the help of non-covalent interaction, while heme can bind to iron atom by the help of coordinate bonds generated by nitrogen in the heme ring. Is it too microscopic for you to understand the importance of bonds? All of these diagrams are the textbook diagram. Sometimes we talk to ourselves that these are the typical textbook thing. So let's take another example. The condition which we face in our daily life. When we shake our hand to somebody, the atoms present on the surface cells of our hand do make chemical interaction with the atoms present on the surface of the second person. Similarly, when we walk on a floor, our shoe surface atoms do make interaction with the surface of floor or road. If the floor is wet, this interaction can be weak. Now our shoe surface make the interaction with the water molecules. Among the water molecules, the interaction is not so strong. So that's why on a wet surface, we tend to get slip. Do you know about Gecko? Gecko is a member of lizard family, and this small cute lizard is very well known for its sticky abilities. It can stick on very flat surfaces like glass with a great attachment power. When we analyze microscopically the pad on the feet, the surface touching region on the feet of these geckos, we discovered that these are minute fibers, hair-like fibers, and these fibers make large number of interaction with the surface molecules. Although these interactions are very weak in nature, but when weak interactions are abundant in quantity, the cumulative force makes the difference. Now let's understand the diagram of relationship. Although this diagram is generated to define the interaction between two molecules by the help of van der Waals force. At large distance, atoms do not feel any kind of attraction or repulsion between them. But sometimes, due to random motion or some other kinetic property, when molecules come closer, they can feel a positive interaction between them and they can form bonds. This positive interaction is known as van der Waals interaction. But there is a limit. How much closer these molecules can come? If the molecules come very close, the attraction force will become a repulsive force and molecules can repel each other. So there is a balance between the attraction and repulsion, the point at which maximum attraction is present and minimum repulsion between the two molecules. This distance is known as the van der Waals distance between the atoms. If this distance is reduced, the power of attraction will become repulsion and molecules will repel each other. So now, do you understand why we call it as diagram of relationship? In relations also, we love our guest, but not for too long. We enjoy our relatives only when we meet them occasionally. If we start living together, you will see that we will generate some misunderstandings or some discomforts and start fighting. The importance of this distance and relation we also feel in our other relations. Sometime we meet someone and we start liking him until the day we start living together. As the distance between the two molecules decreases, the attraction becomes repulsion, and this is the reality of life. When atoms interact with each other, the role of energy is crucial. Some molecules, when joined together, for example, two hydrogen atoms, if joined together, they release 436 kilojoules per mole and form hydrogen molecule, H2 molecule. If we break this H2 molecule, we have to provide the same amount of energy, that is 436 kilojoule per mole, to get these molecules separated. This energy is also known as bond energy. So every kind of chemical interaction has a particular bond energy. For example, in ionic interaction, the energy involved may be 400 kilojoules to 4000 kilojoules per mole. For example, in case of NaCl. If we have a big crystal of NaCl, can we crush it by our hand? So it's very strong. But when we place this crystal into the water, both the sodium and chloride ions get automatically dissolved and separated from one another. This is the most important property of ionic bonds. Ionic bonds are very strong when they are present in anhydrous condition, that is absence of water. But if we provide water, the bond energy is dramatically reduced. But this is not true with the covalent bonds. For example, if you pick a wood piece, the amount of energy required to crush the piece is same whether it is in water or outside water. Covalent bond energies do not depend on the aqueous environment, and they also have a very large quantity of energy. Similarly, metallic bond energies are also very high, but as we are studying about the life sciences or biology, in biology, ionic and covalent bonds are the most important ones. Other than ionic and covalent bonds, various other non-covalent bonds are present. For example, iron dipole bond, hydrogen bond, dipole-dipole bond, ion-induced dipole, or dipole-induced dipole bonds, or dispersion or London dispersion forces. Of all these bonds, London dispersion forces are the weakest one. Although they are weakest, but they are very important in biological assemblies, especially formation of lipid bilayer molecules or folding of proteins. These dispersion forces are very important. If the information is too complicated, let's try to understand in a simple table. In aqueous condition, covalent bonds have energy of 50 to 110, ionic bonds have energy to 3 to 7, hydrogen bond 3 to 7, hydrophobic interaction 1 to 2, and van der Waals interaction 1. So in this table, we can clearly see that covalent bonds are the strongest one, while the non-covalent bonds are weaker. Of these, van der Waals interactions are the weakest. Don't go on the energy because covalent bonds are used in the polymerization. But when protein folds or DNA form double helical structure, or an enzyme interact with a substrate, or an antibody bind to antigen, or a signal molecule bound to a receptor, all kind of reversible reaction in life science depend on non-covalent interaction. Even in your body, all the cells are joined together by the help of extracellular matrix protein and cell-cell interaction. All these kind of interactions depends only on non-covalent interaction, which are weak interaction. So if we remove all the weak interactions, that is non-covalent interaction from the body, our all cells will disintegrate. So to summarize, although covalent bonds are the strongest bond, but the architecture of life is governed by non-covalent weak interactions. This is revision number 2 for torsion angles of biomolecules. Revision is very important. It gives us advantage so that we do not get surprised during our examination, and it also gives us advantage to recall all the important concepts from a topic so that we can score more marks in our examination. Let's start with human posture. In our daily life, while sitting, sleeping or standing, we use different kind of conformation or posture of our body. Some of the postures are correct for our health, while others are not. Human posture is a result of flexibility and rigidity provided by our backbone, that is vertebra or spine. The bones coordinate with the muscles and they are responsible for any kind of posture which we use in our daily life. Now let's see some photographs. For example, this porcupine and this rhinoceros and a beautiful lady. What these three images have in common, which is scarce or absent in this image? You very well know that we are talking about hairs. Hairs are made up of proteins, and these proteins are known as keratin. Keratins are responsible for our hairs, nails, horns, hoofs, and all other similar structures. Proteins come in wide variety of structures, and these structures can be classified at secondary level like alpha helix, beta sheets, turns, loop, random coils, or at tertiary or quaternary level in the form of motifs or domains. What is the property of protein that is responsible for folding of polypeptide into variable structure? Protein is the final three-dimensional folded form. Protein is made up of polypeptide. Unless it is properly folded, polypeptide cannot be called as protein. So polypeptide is the key structure. But like human backbone, polypeptides also have a backbone. For example, in this particular diagram, we can see how two amino acids join together by the help of condensation reaction forming a peptide bond. This peptide bond is responsible for the polymerization of amino acid into polypeptide. Similarly, one water molecule can be added to this molecule and the peptide bond can be hydrolyzed. So the polymer formation reaction is condensation, removal of water molecule from the two amino acids, and breakdown is hydrolysis by the help of water. So to get a better idea of concept of backbone in protein, let's make a structure. This structure is made up of many amino acids. All these amino acids are joined together by the help of peptide bonds. Peptide bond is present between the carbonyl carbon and amide nitrogen. So there are overall 3 peptide or amide bonds. What about the backbone of this polypeptide? Which bonds are responsible? So let's remove all the other bonds which are not the part of backbone. Now this diagram represents the bond present in the backbone of a polypeptide. To make things easy, each of these bonds is represented by a Greek alphabet phi, psi or omega. The first bond between the nitrogen and alpha carbon is known as phi. So every amino acid has one phi bond. Second bond is psi. It is between alpha carbon and carbonyl carbon. So again, every amino acid have now 2 bonds, phi and psi. The peptide bond is represented by omega. So the backbone of the polypeptide chain is represented by a repeated order of phi, psi and omega bonds. Now let's talk about the conformation of proteins. Of these 3 bonds, only 2 bonds are flexible, phi and psi. These bonds show typical single bond character, while the peptide bond represent 40% double bond character. That's why peptide or omega bond is not flexible and is not considered as torsion angle. Torsion angles are also known as dihedral angles or rotational angle. When we study Ramachandran Plot, which represent the ability of peptide to attain different peptide conformation, and this can be represented by the Ramachandran Plot. This plot is all depend on variable phi and psi values only. So whatever the peptide structure is present, alpha structure, beta sheet or whatever, this structure is only dependent on 2 dihedral or rotational or torsion angle. that is phi and psi. Now, do you recognize this lady? This is one of the greatest scientists in our time, Rosalind Franklin. She is known for her work in X-ray crystallography for DNA molecules. Although at her time, DNA structure was not known, that's why this image was a mystery at that time. But later on, thanks to Watson and Crick, the structure was resolved, and now we very clearly know that DNA molecules or RNA molecule can also represent a different type of conformation at primary, secondary, tertiary or quaternary level. So like polypeptide, DNA molecule or RNA molecule should also have a backbone. This backbone is responsible to different kind of DNA molecule conformation like aDNA, bDNA, cDNA, ddNA and zDNA. Please do not confuse cDNA with complementary DNA. In complementary DNA, small c is used, while this capital cDNA is a variable form of DNA conformation. In DNA structure, the backbone is made up of phosphodiester bonds and sugar molecules. So the conformation or torsion angles must be the part of sugar phosphate backbone. These can be again named by using Greek symbols alpha, beta, gamma, delta, epsilon and zeta. Although there are 6 bonds in the sugar phosphate backbone, but there is another 7th torsion bond between the carbon number 1 of the ribose sugar or deoxyribose and the nitrogen present in the base. This chi bond is not directly present in the sugar phosphate backbone, but it plays a very important role in anti and syn conformation of nucleotides. The syn conformation have more steric hindrance as compared to anti conformation. Therefore, in biological system, anti conformation of nucleotide is used. In the biological system, anti conformation is more favorable as compared to syn, and this is the conformation of nucleotide which is also responsible for correct base pairing of adenine to thymine, cytosine to guanine. So therefore, although the last 7th bond, chi, is not present directly in the sugar phosphate backbone, but plays a very crucial role in the final conformation of the DNA molecule. Now comes the carbohydrate. Whenever we studied about carbohydrate, we have this idea that carbohydrates are important source for energy and they are also responsible for our blood viscosity. But carbohydrates are also playing some structural role in nature, like in plants, cellulose, and we use this cellulose for our cloth also, for example while using cotton cloths. And carbohydrate also plays structural role in our human body in extracellular matrix in the form of glycose, aminoglycans, proteoglycans. So the structure of starch, glycogen and cellulose, these all represent the polymer of glucose molecules. Although starch is composed of 2 different types of polymers. One is amylose, made up of glucose molecules joined together by alpha 1,4 glycosidic bond in a linear fashion, while amylopectin is a branched structure. At the end of every branch, there is a non-reducing end, and which is the target place to remove glucose molecule from these polymers. Starch is a storage carbohydrate in plants. Similarly, in humans and other animals, glycogen play the same storage role. Here, the structure is very similar to amylopectin, although it is heavily branched, much more branched as compared to amylopectin. And the reason is that because all the free glucose will be removed from the non-reducing part, so being animal, we need a quick storage form of carbohydrates from which we can easily and quickly remove large number of glucose to produce metabolic energy. Cellulose play important role as a structural molecule, and these are also made up of glucose molecule, although glucose is not alpha. Here, the glucose molecule is beta type. These are linear molecules which are joined together by the help of hydrogen bonds between the glucose molecules. So as with the other polymers like nucleic acids and proteins, polymer of carbohydrate can also show different conformation. For example, amylose generally present a helical shape. Indeed, alpha amylose helical structure is the first ever biological helix discovered. And also, the alpha helical structure of amylose is the key player in generation of blue color when starch is treated with iodine. When two molecules of glucose join together, they are joined by the help of glycosidic bond. And these glycosidic bonds are very crucial for the conformation of carbohydrates. Therefore, these bonds are considered as torsion bonds or dihedral angles in carbohydrates. As previously, these bonds are symbolized by using Greek alphabets phi and psi. So when carbon number 1 is joined to carbon number 4 of any other sugar molecule, there will be two dihedral bonds, that is phi and psi. But in branching, where carbon number 1 is joined with carbon number 6 of glucose molecule, there will be three torsion angle phi, psi and omega. Here all the three bonds are flexible and all these three bonds are responsible for the final structure of the polymer carbohydrates. Therefore, it should be very clear, like protein and nucleic acid, carbohydrates can also show different conformation due to the torsion angles present in their polymeric form. Let's summarize the information we just learned. Protein have two dihedral angles, phi and psi. Although third bond is present, omega, which is known as peptide or amide bond, but due to its 40% double bond character, these do not provide flexibility to contribute in the final conformation of the proteins. So for proteins, only two dihedral angles are there. In nucleic acids, specifically DNA, there are 7 torsion angles, of which 6 torsion angles are present in sugar phosphate backbone, while the 7th chi torsion angle is present between the sugar and base glycosidic bond. And it is responsible for the anti and syn conformation, and that is also crucial for the final structure of DNA or RNA. So DNA has 7 torsion angles. In carbohydrates, at least 2 dihedral or torsion angles are there, phi and psi. In some cases, like in branching points where carbon number 6 is involved, 3 dihedral angles are present, phi, psi and omega. Here all the 3 bonds are single bonds. Therefore, all 3 bonds contribute to the final structure of carbohydrate. What about lipids? Lipids are considered as 4th biomolecule class. But in typical sense, lipids are not typical example of polymers. Phospholipids can join together to form lipid bilayers. Lipid bilayer is the polymeric form, but it is not a true polymer because phospholipid molecules are not joined together by covalent bond, but they are joined together by the help of hydrophobic force. Therefore, we generally do not consider torsion angles in the final polymeric form of plasma membranes.